I'm Matthew Wood. I'm Professor of Neuroscience at the University of Oxford. My research is focused on the development of new types of treatments for neurological diseases, one of which is Parkinson's disease, and that's why I'm delighted to be here to talk this evening. I started out as a medical student, and I became a neurologist, and I saw lots of patients with untreatable neurological diseases like Parkinson's disease that 20 years ago we had no prospect of doing anything for, which is why today I spend most of my time in the lab really with a major scientific interest of trying to develop treatments for a whole range of neurological diseases such as Parkinson's. I think to divorce the scientific work from the patients, who are our ultimate goal to cure the patients, would be impossible. So every possible opportunity to welcome people into the lab and to explain what we do is something we grab hold of. Parkinson's disease is a complex disease, and this is not going to be solved. A cure is not going to be found by one research group, by one institution, even by people working in one country. It's a vast international collaborative effort. It will represent a major step forward scientifically for countries and individuals, governments, charities, collectively.
What I really want to talk about in the next 30 minutes is not necessarily how we would develop a new type of medicine to treat Parkinson's, but I want to tell you about the importance of how we can think about delivering that medicine to the right part of the brain. Without the ability to deliver it to the areas of the brain that are affected, developing the medicine is unlikely to have any benefit. And hopefully I can convince you of that in the next 30 minutes. So Mark has very helpfully told you quite a bit about the brain and its organization and complexity. I just want to repeat that for a second, but to give you a slightly different angle on it. Here we're looking at two areas that are implicated in Parkinson's disease. Here is the midbrain, a normal midbrain and a midbrain from a patient with Parkinson's, and you can see the grey cells here are no longer present here. And then a related area to which these neurons project. This is the putamen, a healthy putamen, and one that is now a bit smaller because it's undergone degeneration. So the message here is that Parkinson's, and from this scan image you can see it very clearly, involves more than one part of the brain. And Mark has told you that it involves many different cell types or variations of dopamine neurons within this part of the brain, but it involves more than one part of the brain. If you then think about some of the different features of the disease as it develops, you will appreciate that it involves even more areas of the nervous system. So we talk about some of the premotor symptoms that patients with Parkinson's get, and these may be related to, for example, the gut, constipation. Patients often develop problems with smell. These often precede any problems to do with walking or locomotion. And then, of course, often as the disease progresses and towards the later stages of the diseases, there can be cognitive effects that come on. And these all relate to different parts of the brain, whether it's the cortex, the midbrain that I've talked about, the olfactory areas to do with smell, or even some of the neuronal connections and neurons that control our gut. So therefore, thinking about how one would deliver a medicine involves not just thinking about the dopamine neurons in the substantia nigra that Mark told you so eloquently about. It will involve thinking about all of these interrelated areas and how we can best deliver medicines to all of these areas. So if we were to think about what an ideal kind of treatment might look like, I think that treatment, and a treatment to prevent Parkinson's, which is what we really want ultimately, it's a treatment that is going to have to be delivered in a systemic way to the body in order to reach many different parts of the brain and even to reach, for example, the gut. I think injecting a treatment directly into the brain may have some benefits, but it's unlikely to be the answer ultimately, unless you inject it in multiple sites and repeatedly over many years, which involves surgical intervention and is probably not desirable. So we need a treatment that can reach all the parts of the brain and the gut, for example, that are affected. And ultimately, we also need that treatment to be administered pre-symptomatically before patients have developed symptoms so that we can actually prevent the onset of symptoms. So those are two enormous challenges. And I'm going to spend most of my time talking about the delivery aspect, but I'll touch on this towards the end of the talk. So the major obstacle to being able to deliver a treatment systemically is the presence of a structure called the blood-brain barrier, which, if you like, encircles the brain and encapsulates it or walls it off from the rest of the body. And as a result of this, the barrier, or our ability to get things into the brain, is diminished greatly. And surprisingly, even to those of you who may know something about the blood-brain barrier, this barrier is a major barrier to both very small and large drugs. So I just want to make this point quickly in passing. The small drugs are the classical types of drugs like aspirin or paracetamol that we all take, that have been developed by pharmaceutical companies over many years. But surprisingly, only about 5% of these drugs actually are used for treating brain diseases. And that's because the vast majority of these drugs do not penetrate into the brain, even though they're very small molecules. Surprisingly, and rather a good thing too, one of those drugs, one of these very small molecular drugs that does get into the brain, is one that you'll all know about called L-DOPA. But most small molecule drugs don't get into the brain. Of the types of drugs that pharmaceutical companies are now developing, these are the very advanced new types of drugs, which we call large molecular weight or biological drugs. These are proteins, these are antibodies, these are big drugs. Of the top-selling drugs in 2014, 8 out of 10 of these are these biological drugs, big drugs, and none are used to treat neurological diseases. And that's because they do not get into the brain. So currently, our ability to take advantage of all of this new wealth of knowledge about drug development is impossible unless we have a method to deliver these into the brain. And just to make this point again, but in a slightly different way, about 98% of all small molecule drugs do not get into the brain. About 100% of all the big ones don't get into the brain. Most drug companies don't have any research that's involved with delivering drugs to the brain. And most academic groups don't think about this either. Now, this is not entirely true. I'm slightly exaggerating, but this is also not entirely true because this was data taken from 2010, and surprisingly, even in the last 3 or 4 years, things have changed, and I'll come back to that point as well. But this really illustrates the magnitude of the challenge. Not only do most drugs not get into the brain, pharmaceutical companies and academic research groups have not historically been devoting the effort and interest into trying to solve this problem. And in fact, one of the world experts in understanding the blood-brain barrier itself has said this not long ago, 3 years ago, that any attempt to develop a drug for brain disease, a neurotherapeutic, by concentrating only on the drug and not giving appropriate emphasis to delivery is highly likely to end in failure. And I would agree with that. So I think there needs to be a fundamental change in our thinking in order to try and solve this problem. So what is the blood-brain barrier? This is Paul Ehrlich, a very eminent, famous German physician-scientist of the 19th century, who was the first person to describe the blood-brain barrier. Now, I haven't been able to get you the exact images that were reported in his research, but what he did was inject a small dye into the bloodstream, and he could detect that dye in every single tissue in the body except for the brain, which told him that there was some barrier that existed. Now, I haven't got that picture exactly, but what's been done here is a small molecule blue dye called toluidine blue has been injected intravenously, and most of the brain here is completely white. In other words, the dye hasn't penetrated into the brain. It has penetrated into this area here, and that tells us that the blood-brain barrier around this part of the brain was probably leaky. Okay? So he described this barrier something like 100, 150 years ago. We've known about it for a long time. The barrier is essentially a barrier between the blood vessels that permeate the brain and supply it with blood and oxygen and all the nutrients the brain needs, and the brain tissue itself that Mark was talking about, all of the brain cells. And here just shows you the density of the blood vessels in a part of the brain cortex, an incredible density of blood vessels throughout the brain. And between each one of these vessels and the brain parenchymal tissue is this barrier. In fact, the vessels are so dense that every brain cell in the brain that Mark was talking to you about is probably within one brain cell's width of a blood vessel. So they sit right next to blood vessels, not surprisingly, because they need all the oxygen and have all the metabolic requirements that are satisfied by that blood supply. So they sit in close proximity, but between them and the blood is this barrier. So what constitutes the barrier itself? Now, if you have a look up here, we are looking at, in this little cartoon image, very high magnification of a blood vessel, a blood capillary, as it passes through the brain. And this is the blood vessel here. It's surrounded by a single cell called an endothelial cell, underpinned by something called a basal lamina, but two other important cell types, a pericyte and then an astrocyte. And the astrocyte is one of the glial cells, which is one of the supporting cells of the brain. So you can see that the barrier here is comprised of cells that are joined together very tightly and together function as a barrier, preventing the influx and efflux of substances from the blood into the brain, and in particular all of the drugs that I mentioned. Now that's actually not entirely true. The barrier is something we call a barrier, but we now know that there is a lot of flux and a lot of travel across this barrier all the time. So it's paradoxical in a way that we can't get drugs readily across it, but all of the material requirements of the brain readily cross the barrier. And there are a number of different mechanisms by which water-soluble agents and lipid-soluble agents, etc., get across the barrier, but important things that your brain needs readily get across the barrier, such as sugar and amino acids, and even other larger things, such as insulin and other proteins. And interestingly, insulin and other proteins have highly specialized ways of getting across the barrier. They bind to things called receptors, and they get readily transported across the barrier, and they get ejected or dumped out onto the brain side, and the insulin or the protein is then within the brain. So we now know there are highly sophisticated transport mechanisms across this barrier. In fact, it's hardly a barrier at all if you happen to be insulin or if you happen to be an amino acid. So how should we think, and how can we think about delivering drugs more effectively across this barrier? Well, not surprisingly, since we've known about this barrier for an awful long time, there's a long history of people attempting to understand how this could be done. Many, many methods have been tested and tried. So these include some of those listed here: the use of lipids, the use of insoluble fats as a way of delivering drugs, the use of small peptides or proteins as a way of delivering drugs, and the use of biological agents, for example, things like viruses, as a way of penetrating from the blood into the brain. And most of these have been pretty unsuccessful to a greater or lesser degree, but nothing remotely successful enough to be thought of as the answer to the problem. However, there is increasingly, and particularly actually since 2010, as I've said, increasingly a lot more attention focused on this problem. And so one thing I'm not going to talk about tonight is the work that's been done over the last few years to utilize antibodies as a method, as a technology for delivering drugs across the blood-brain barrier. So currently, at the moment, this is probably the favored method that pharmaceutical companies are hinging all their hopes on as a technology to deliver drugs into the brain. And it does look promising. But what I want to tell you about is something completely different that we think has a lot of potential and that I hope ultimately one day will prove to have huge potential. And these are things called extracellular vesicles. So what is an extracellular vesicle? And if you'd asked me that question 10 years ago, I wouldn't have had a clue. I've been working in this area for 20 years, but this is something that's relatively new to me as well. Here's a picture of some of these vesicles. This is under the electron microscope, so they're actually very small. So this research paper was published in 2007, and this really certainly changed the way that I think about the problem I'm talking to you about, but it changed many people's thinking about this problem. So this is from a wonderful research group based in Sweden, who made the astonishing discovery that these vesicles, these small little fat droplets, if you like, tiny fat droplets, are released from every single cell in our body. And that they contain important information in them, such as protein, but also genetic information. And that their function is to convey those bits of information to other cells in the body. So a communication system for transmitting large proteins and large bits of genetic information had been existing in our bodies, and we didn't know it was there. And it transmits this information between cells in our bodies, which seems remarkable that we didn't know about it. We immediately thought, amongst many of our other thoughts when we first learnt about this 7 years ago, doesn't this seem like exactly the right type of thing that you'd need if you wanted to deliver a protein drug or a bit of genetic information as a drug, as a treatment? Wouldn't it be wonderful to utilize the natural system that exists and that has evolved over millennia to do exactly this? And so that's what we set out to try and do a number of years ago. Now, in the last 10 years, we've learnt an awful lot about these small vesicles. They're also called exosomes, so if I use that term, they're interchangeable with the term extracellular vesicle. So here schematically showing you a number of these types of vesicles. They vary in shape and size, but they're all very small, and so we really could term them nanoparticles. They are a type of nanotechnology, if you've heard that term. They're very small. We now know that they play a fundamental role in many of our bodily systems and functions. They play a fundamental role in our immune system, in regulating blood clotting, in regulating the activities of the stem cells in all of our body and all of our tissues, and even in repairing our tissues. And increasingly, we know a lot about how these vesicles can play a role in disease. And this is particularly true of cancer, where we know an awful lot about how important these vesicles are. So they transmit this important information between cells, and we now know that this flow of information is absolutely critical to the way that our bodies function in a normal, healthy state. So the question really that we asked ourselves about 7 years ago was, could we harness this ability of these vesicles to transmit information, to think about a completely new way of delivering larger drugs, protein and drugs that might be genetic information, to the brain. And I was very fortunate to have working with me at the time two fantastically talented people, Ichi Sao and Lydia Alvarez. Ichi is from Singapore and Lydia is from Spain, and together they set about working on this project. Here is a picture of one of these vesicles. What we first had to do was to develop the methods to isolate these vesicles so that we could gather them, isolate them, purify them, and know that within a test tube we had a nice pure sample of the vesicles that we could use in our work. The second thing that we did was to think about ways in which we could modify these vesicles to encourage them to travel towards the brain. And so what we did was to insert into the membrane of these vesicles a peptide, a fragment of a protein called rabies virus glycopeptide. Now, This maybe doesn't sound terribly good to you, as it's got the word rabies and virus in it, but it's a small fragment of a viral protein that is the critical bit that the rabies virus uses to get into neurons, to get into the nervous system when that virus infects the nervous system. And so what we simply did was took the useful bit, which is shown here, the peptide sequence, and stuck it on the outside of these vesicles. The only other piece in the puzzle, really, was to develop a way of putting our drugs inside the vesicles. And I would have to be honest and say we far from perfected this method, but we've been able to do this to at least some degree of efficiency. And so what I'm showing you here is encapsulated within that vesicle a small piece of genetic information, which is a double-stranded piece of RNA. And you need not worry too much about what that is, but it's an important piece of genetic information, and that increasingly is being developed as a drug. Not for brain disease, of course, but for other types of disease. Now, since Lydia and Ichi did their work in the years 2007 to 2011, many other people have been involved in the work in the lab, and I won't tell you about them all. Although Samir Andalusi, who's now based in Stockholm and was working with me for some time, has been really critical to doing all of the things I'm going to tell you about. So here you can now see an image on the left-hand side where we have injected these vesicles intravenously into a mouse, and we've labeled them in such a way as we can detect them with a camera. And what we're looking at here under the camera is the brain. And you can see this is the brain from a mouse that hasn't been treated. This is the brain from a mouse that has been treated with the vesicles, but without the rabies virus peptide attached. And this is the brain from the mouse that has been treated with the vesicles with the rabies virus peptide attached, showing you that there is really very easily detectable penetration of these vesicles into the brain. There is also very clearly detectable drug within the brain. Now, this may be less clear. We're now looking deeply into the cortex of the mouse brain, and the drug, the RNA drug, is labeled up in red. In the green, these are all labeled up as neurons in the mouse cortex. But you can see, if you can see, look carefully, every single one of these neurons has red dots, which tells us that this bit of genetic information, this genetic drug, has been effectively delivered to just about every single one of the neurons that you can see in this image. So not only has the delivery agent, the vesicle, penetrated into the brain effectively, the drug itself has been delivered to the neurons, to the brain cells, and we can detect it on an image like this. So this was really remarkable when we first saw this. This was astonishing because it wasn't something that we'd expected. We thought this would be almost unachievable, and to have done so within a relatively short time was really remarkable. But this is not the real question in this case. The question then is the drug active? Does it do anything? Having successfully delivered it, having successfully crossed the blood-brain barrier, is this drug active? And so this was information that was in the first publication that we produced in 2011, showing that this drug was active. The genetic drug was designed to switch off a gene that is important in Alzheimer's disease, and that's what we were looking at the time, 4 years ago. The drug's called BACE1. And you can see here is the untreated mouse, and here is the treated mouse, and we've been able to switch that drug off by about 70%. This is looking at the RNA, and this is looking at the protein. They're both about a similar degree of silencing, as we call it. So this gene that is implicated in Alzheimer's disease, we'd managed to turn it off by about 70% within the brain by utilizing this method for delivering these large, complex genetic types of drugs into the brain. We were really surprised and pleased that we could see such a positive effect. And of course, that has launched us into all sorts of other developments with trying to understand how to exploit this technology. And of course, that's when we were delighted that Parkinsons UK wanted to get involved and to help us develop this work and this technology for Parkinson's disease. Now, in the case of Parkinson's disease, it might be slightly unclear to those of you who know something about the disease, which would be the gene that you would go for? Which would be the gene that you might try and switch off? And to some extent, that doesn't matter for us now, because what we're trying to prove is that we have a method for delivering these complicated drugs. But the gene, the obvious one, really, is the gene called alpha-synuclein. As you know, one of the pathognomonic, one of the characteristic features of Parkinson's in the neurons of the brain is the development of these structures called Lewy bodies, these aggregates of protein. And one of the principal protein constituents of these aggregates is alpha-synuclein. And although we don't know exactly what these aggregates of protein do, we believe that they're bad. And therefore, reducing the levels of alpha-synuclein, and particularly the levels of alpha-synuclein within these Lewy bodies, would seem like a very good thing to do. And so this is what we set out to do a couple of years ago. And to cut a long story short, we've got some very encouraging results that lead us to believe that we're going in the right direction. So for alpha-synuclein, we're able to switch off alpha-synuclein, and here to a degree of about 50%. So here are the untreated mice, and here is the treated mouse. There's about a 50% reduction in the level of alpha-synuclein using the vesicles to deliver the drug. And this is true in 3 different parts of the brain: the substantia nigra, the striatum, and the cortex, which reiterates the point I made right at the beginning of the lecture, which is that we do need a method that allows us to deliver our medicines to multiple brain areas, all of the areas that are affected in Parkinson's. And what's more, we are now able to see a reduction in the numbers of aggregates within the brain. So here is the control brain, where the aggregated alpha-synuclein protein aggregates are visible, and here is the treated brain, where we can see a very obvious reduction in the level of aggregation. So this is the first very encouraging data. It's not published yet. And I should say this now is a very, very important collaboration between 2 of the people that we have trained in our lab in Oxford and that we work closely with. Lydia, who did the original work, is now based in London. Samir, who did some of the subsequent work, is now based in Stockholm. And so between us and London and Stockholm, we're working very hard on this problem. And it's credit essentially to Lydia and Samir that we've got as far as we have. So I think this is extremely promising. But there's a lot more work to do. So I would be very dishonest if I were to stand here and say this is going to be available for use tomorrow. There's a lot more to be done. And I'm just raising a number of the questions that have probably occurred to you already as you've been listening to what I've said. How do these vesicles get into the brain? We don't know. What is the best way to get them into the brain, and what is the best source of these vesicles? This is something we could discuss, but we're far from knowing the answer to this. Most of our efforts at the moment are focusing on stem cells and utilizing vesicles that have come from stem cells. We believe for many reasons they are probably the best vesicles. Can these vesicles deliver other types of drugs? The evidence I've shown you very briefly is that they could deliver a genetic drug. But we now know they may be useful even for small molecules, the 95% of small molecules that can't get into the brain, and genetic drugs, and proteins, and even antibodies. So they could be very useful for many different types of drugs as a way of delivering these effectively into the brain. And what's more, there really is a fundamental change in the way people are thinking about delivering drugs to the brain from even 4 years ago, such that we now have a vast number of partners within Europe that we're working with to develop this type of technology. A number of academic groups, but even 8 pharmaceutical companies are interested in doing this. So there's been a sea change in understanding the importance of being able to deliver drugs into the brain, and many more people now, rather than 4 years ago, are interested in doing this. Now, I just want to raise 2 final points, because they're important to think about in parallel. The one is when to treat, and I've said ideally a treatment like this that would aim to deliver a preventative treatment to patients, by definition, would need to be delivered before patients develop symptoms. And so Mark very eloquently told you about dopamine neurons, that the numbers of these decline over time in Parkinson's, and that only when a certain critical threshold is reached do patients begin to develop symptoms. And therefore we need somehow to discover before these symptoms develop, the patients have Parkinson's. And so it's this preclinical phase, before patients develop symptoms, that is going to be absolutely critical to be able to identify so that we can apply these preventative treatments at the right time. And you will have heard this term before, but the identification of biomarkers, whether they be something that you could look at on a CT scan or an MRI image, or something you could measure in the blood, is going to be very, very important to allow us to identify the right time to administer these kinds of preventative treatments. If we're left to do it down here as the disease develops, they may not be nearly as effective as they would likely be earlier on. And then one final thought to leave you with. Many of you will know, and you might have appreciated from what I've said, that because many brain areas are implicated in Parkinson's, there has been discussion, particularly over the last 5 years, about how Parkinson's disease spreads through the brain. If the initial symptoms begin in the gut or the olfactory parts of the brain, and the final part of the brain to be affected is the brain cortex, does the disease actually spread through the brain? Here it's shown schematically. The different areas that become implicated in the disease grow with time, and different brain areas become involved. What causes this spread of disease? Is it just simply that there is some differential susceptibility of different parts of the brain? In other words, the dopamine neurons are more susceptible down here than the cortical neurons are up here, or is there some other factor involved? One of the intriguing possibilities that we, and actually a number of other laboratories, are investigating is the possibility that the spread of Parkinson's is via the very vesicles that I've been telling you about. So Yoshi Aoki, a wonderful Japanese scientist working in my lab, is now investigating this very intensively, as many other people are, the possibility that the vesicles that we've been exploiting to deliver drugs to the brain are themselves the method by which the disease is conveyed between cells. We know dopamine neurons generate lots of these vesicles. We know they're produced within the brain. Could it be that they are actually involved in transmitting the disease between cells in the brain? This is very speculative at the moment. It may or may not turn out to be true, but it's a very interesting fundamental problem that I think if we can solve it, may well even lead us into understanding how we can exploit these vesicles better for treatment purposes. So really what I've told you about is a new way, we believe, to solve this problem of how to deliver drugs, complicated drugs, big drugs particularly, to the brain by the use of a natural transport system that does just that within our bodies. And I've also hinted at the possibility that these vesicles may be involved in the development of diseases like Parkinson's, and therefore understanding what these vesicles do in both health and disease is going to be very important over the next few years. So that's where I want to finish. I couldn't have done this without all the members of my lab, but particularly we couldn't have done the work I've told you about without the support of Parkinson's UK and the many other funding agencies who helped us to get started over the last 10 years. Thank you very much.